Yes, hello, everyone. It's my pleasure to welcome you to this first of the e-Symposium series for IDG. And since time is short, without further ado, it's my pleasure to introduce our external speaker, Dr. Ian Dunham. He is director of the Open Targets project, which is hosted by EMBL-EBI. Open Targets has a number of strong industrial relationships, and although it seems like we are in competition, we are actually very much trying to work in synergy. So welcome, Ian, and thank you for agreeing to be the opening talk for this symposium.
Great, thanks. So I'll just share my screen and hopefully going in presentation mode now. Can you see that?
Yes, we can.
Great. Okay, so I'm going to talk to you a bit today about the informatics tools that we've been developing at Open Targets and what we've done recently, mostly in the last year, in taking those further. And the way this talk will be structured, so I'll just give you a little brief introduction to Open Targets, then talk about genetics evidence and how we use genome-wide association evidence through a tool that we call the Open Targets Genetics Portal. And then a section on the new developments that we've made in the Open Targets platform, which is our main tool for target identification and prioritization. And then right at the end, just a brief summary of where we think we'll go with that next. So to start with, on Open Targets, as I think you'll know, making the choice of the target that you want to use in a drug discovery program is a critical point in the process, because pretty much everything that you do downstream of that is dependent on that target decision. And some years later, you'll find out whether that decision was correct in terms of success in clinical trials. One thing that we do know is that genetics information is a predictor of progression or success through the clinical trials process. And if you have a target with genetics information, you're about twice as likely to go through the phases in clinical trials than if not. And so it's important to bring that causal evidence to targets when you're at the decision process. Open Targets is a partnership which aims to use genetics and genomics to transform drug discovery and enable systematic identification and prioritization of targets. We concentrate on those high throughput systematic approaches, and we bring together the Sanger Institute and EMBL-EBI as our two academic partners, and then currently GSK, Sanofi, and BMS as pharma partners. And we work closely together in teams on individual projects. And I think probably if you're listening to this talk, you'll know Open Targets platform and the publicly available tools that we make. But we also generate a lot of experimental data as well. In specific disease areas, so in neurodegeneration, in immunology, inflammation, and oncology. And so our approach is really to bring together this experimentally generated information with the publicly available data to provide evidence that a target is involved in disease. That's really our key focus, is on this relationship between target and disease, because that's predictive of where you go downstream of that. And we want to build a therapeutic hypothesis around that information. We really focus on target and disease and the evidence, but also we pay attention to drugs, obviously, because they're an indicator of success when you have successful drugs in clinical trials. And so we also, in the model that we use, we think about the mechanism of action and the indication that's being targeted by those drugs. And so we're trying to sort of square this or to bring together this triangle of how does the evidence on the association of a target and disease lead to a drug? And can we use existing drug information to shed light on the success or the validity of the various evidence sources that we have? We use a range of different evidence sources for this relationship between target and disease. A lot of it is strongly genetics influence, information coming from sources like ClinVar, from Genomics England, from our own Open Targets genetics portal. We have specific data streams in cancer from the COSMIC database, and also Intergen on cancer driver genes, pathway information, specific experiments that we and others have done in systems biology or in CRISPR screening, but also then mouse models and literature information as well. And we bring all of those together in the Open Targets platform, which I'll talk about in the latter half of this talk. But what I want to focus on initially is Open Targets genetics and how we bring genetics information into this equation, particularly for genome-wide association data, of which there's a very large amount. So I'm going to talk to you now about Open Targets genetics. The URL is genetics.opentargets.org. Over the last 10 or 15 years, there have been thousands of genome-wide association studies published linking disease or linking loci with the various diseases and traits across the genome. I've already said that targets with GWAS information are more likely to succeed in clinical trials. So we want to try to marshal this information together. But there's a fundamental problem here in that when you look at the variants that underlie GWAS associations, most of them are outside of the coding regions of genes. So they're not protein coding changes, and you can't directly attribute them to the nearest gene of interest. So there's a problem about how do you link the association between the variant that you pick up the association with in the first place and the gene that's being involved. And it's a tough process to move across from the initial study through the lead SNP that's identified in the GWAS study, through fine mapping to all the variants that have any association with the disease, and eventually to the gene. But there's a number of tools that one can use here. And what we've tried to do is to integrate these to provide a systematic approach to going from the association to a likely candidate gene. And this is what we do in Open Targets genetics. So we take human genetics for complex traits, we walk through the association and then a series of genetic processes to get the fine mapping for all likely variants. And then we use functional genomics information, looking at whether that variant is associated with a particular disease, a particular gene, is it connected by functional genomics? This gives this in a little more detail. So initially what we did with this was to take all of the GWAS studies in GWAS catalog and also every GWAS that's published in UK Biobank, identify lead variants, and then associate all of the tag variants that are around there that may have some involvement, and then use a series of functional genomics data, including expression quantitative traits, physical interactions like promoter capture, high C interactions, DNase-1 correlation, and also variant effect prediction to link each tag variant to genes. And so you have a path through from the association to the many variants that are involved in the association and many genes at the end. But actually, that's not really what you want. What you want to do is to establish for each locus which is the most likely gene. And so what we've been doing over the last year and a half or so, or more specifically Ed Mountjoy here has been doing, is to develop a locus to gene score that uses a machine learning approach to take each locus train a set of gold standards based on the functional genomics data, and then use that model to predict the most likely gene at each locus. Again, in a little more detail, here's some of the studies that we used from GWAS Catalog, UK Biobank. There are studies that have only the lead SNP, but also summary statistics where you have all you have statistics for the association for all the SNPs that were studied. And then we combine that with functional genomics information, as I said, expression QTLs, promoter capture high C, and so on. And the process that we went through was to take the GWAS signals, identify through fine mapping the lead SNPs, so that every SNP in the across the genome has some kind of probability of being associated with the disease, pick out the variants that are most likely. At the same time, we took every GWAS trait and every molecular trait and performed an analysis that's called co-localization, where we looked to see if the two signals at each locus in the study suggest that the traits are related to each other. We then take all of that information and combine it, so we have a vector of features across all the different data sets that we have, the in silico predictions, the co-localization evidence, the credible set of SNPs, and all the epigenomics information. And we train that in a supervised way on 445 curated causal genes. And this is what goes into the, we use a XG gradient boost model and produce a model that gives a ranking at every locus across the genome for every trait involved, and a score of the likelihood of the genes being involved, which we call the locus to gene score. This score is reasonably predictive. There's the precision recall and the ROC plots for that. We can also test whether we recover literature mined text mined information around each of the loci. Do we recover the genes that are mentioned for these loci in these diseases? And using this model gives an improvement on just using nearest gene alone. It's indicated by the darker purple colors in the plot here. We can also look at known genes across Kemble data, and again using the model that's learnt in machine learning gives us an improvement in identifying or picking up genes with existing drugs. There is one caveat to this, which is that if we also include network information across loci, we can get a further improvement on the score. So this model is well calibrated and predictive. It's an improvement on just using the nearest gene alone. A lot of that improvement actually comes from the way that we use the genetics information to aggregate the signal to give a more specific version of the signal. And as I said, we can get further gains by including network information as well. Looking forward, we need a lot more gold standards because we don't cover all possible, or we don't cover all the different formulations of regions. We need more data across different tissue sets and so on. But I think this is a way to go to improve our understanding of GWAS and get to targets. And all of that is included in genetics.opentargets.org. We're now going to move on to the Open Targets platform, which I hope many of you looked at. In the current version of the platform, we perform a number of analyses and integrations to identify and prioritize targets. So we take a set of evidence sources, genetics, somatic mutations in cancer, known drugs, expression, text mining, animal models, and so on. And we integrate the evidence from different sources into these categories, which we call data types. We also include a step where we integrate the diseases through an ontology that we call the experimental factor ontology. And this allows us to expand evidence up to different levels in the ontology, which means that we can integrate these different data at different levels. We score each of the evidence in a principled way within each data type. And then we score all of the evidence together using a harmonic sum function to lead to an overall association score. We also include various target-specific factors to allow prioritization, such as tractability of the target, safety information, and expression data on each of the targets. Oops. Over 2020, we've made a lot of updates into the annotations that we include in the Open Targets platform. So improvements on our drugs, indications, and clinical trial information from Kemble. We've moved to a new version of the ontology. We continue to expand coverage of chemical probes and target enabling packages from the Structural Genomics Consortium. We've improved the cancer hallmarks from Cosmic, and the target tractability assessment has expanded to various modalities as we go forward. We also include e-tox information and tox21, and updated our literatures for our dictionaries for named entity recognition for these evidence. And this is that's a process that we continue to incrementally improve each of these data sources. In addition, we've added new data sources. So we've included adverse drug reaction information from FAERS. We use the log likelihood ratio to calculate the significance of these data and present that alongside the targets and drugs. We associate each drug with its target. We've also improved our phenotype annotations across all the different diseases. So we've added 8,000 phenotypes across 5,800 diseases using Mondo and the human phenotype ontology. And so this gives a much richer way to look at the various diseases that we include in the platform and the evidence. And every time we do this, we chart how the target disease landscape across our different data sources changes. And so we generate these kind of plots for each of the different data sources. And you can see that there are events that occur where we might expand the number of data. So when we change versions, or we change the way that we use the ontology, or perhaps we initially with genetics, we increase the number of evidences that we have and then reduce it down when we apply a more strict threshold to the data. So we keep track of all of this information as we go along. So now I just want to look at the next generation of the platform that the team have been working on over the last year. So we've completely redesigned and rewritten the platform to try to relieve some of the technical debt that we had in the project. So this has included completely new data modeling of feeds, trying to reduce some of the nested nature of the data so that we were using, to simplify the data, enhance the information in the data sources, and harmonizing the fields so that there was a lot more interoperability. We also completely refactored the data ingestion pipelines for this new structure that we use. That has also then led to a completely new extract, transform, and load pipeline and the back end of the database, which gives us a lot more flexibility and scalability. So we're not tied to idiosyncratic structures in the data. We've also improved various of the scoring algorithms, which will enable us to do on-the-fly calculations very quickly now. So eventually this will lead to being able to reformulate the score, even to apply different algorithms to the score at the user's preference, and it improves performance dramatically. And then finally, we've rewritten the web application. At the moment, we've duplicated the functionality that we have, but the new structure with new API will actually in the future allow us to devise new views of the data. So the new web experience, so it all has the same open targets blue structures, but we now provide the information in a series of modular structures, and you can move these modules around. You can decide what your favorite order is and keep it. We reproduce most of the views that we had before, but making them more refined and smoother to operate. We also provide a lot more detailed evidence because of the flattened structure of the data. We're able to provide a lot more information. Here we're looking at various genetics information. We can get down to a lot of detail in the evidence that wasn't there previously, although it was buried in the evidence somewhere. And then we've enhanced some of the strategies. So previously, when we presented diseases associated with a specific target, because of the way that we structured the ontology expansion, you would get a lot of meaningless terms at the top, for instance, of these scores, which were just aggregated terms. We've improved the algorithm to do this, so we get more relevant diseases at the top of the scoring now. And we've included new data. So we've started to include data from one of our projects looking at integrating various interaction databases, Intact, Senior, and Reactome coming together along with StringDB so that we can show proteins that interact with a target. Here we're looking at ANGPTL3 and seeing the interaction with lipoprotein lipase, because this is very relevant in familial hypercholesterolemia. So we're beginning to have this interaction data. We'll gradually go further and start to be able to expand targets on the basis of these interaction maps. And finally, looking forward, what are we going to be doing in the future? Well, the new structures allow us to be a lot more flexible in the features that we can bring in. One of the main themes that we want to develop over the next year or so is to allow prioritization across a more diverse range of therapeutic hypotheses. So this will allow more modeling of more complex features in the evidence and make them available for different queries. The streamlined infrastructure will allow new data to be ingested more quickly and be used in this way. And what we want to be able to do then is to empower the users to evaluate their own hypotheses, including things like being able to alter the score on the fly, bring their own models into the score. So for instance, one might want to allow the user to show all targets with genetic evidence for involvement in inflammatory bowel disease, for instance, and then to be able to identify the ones that would are predicted to provide benefit through an inhibitor, specifically expressed in the gut, and are tractable by an antibody type drug. So to bring together each of those prioritization and identification features in more sophisticated queries that build to the therapeutic hypothesis. So just to finish off then, just to acknowledge everybody in Open Targets, but also the specific teams that work on the left on the Open Targets platform, and then here on the genetics portal specifically. So David leads the development of the Open Targets platform with the rest of the team. They've done a lot of work over the last year to develop the features that I've shown you. And then Maya, Mode, and Jeremy have been working on the next version of the Open Targets genetics. So I'm going to stop there, and I can answer questions if there's time.
There is one question that has popped into the Q&A. It says, based on your locus to gene scoring work, do you find that the relevant non-coding Snips consistently fall within the standard ranges of distances upstream, downstream from the target gene in question, or is each case unique?
So you can show that there is a distribution that they fall into. So if you look across all the data, they tend to be nearer in general, but I wouldn't say if you looked at any specific one, using distance is not necessarily a very good way to associate that. So it's more about where are the regulatory features for those genes. So you will see variants in promoters, you'll see variants in enhancers, but those enhancers can be distributed both within the gene, but also at distance 5' and 3'. So there's not a specific pattern as such.
Okay, thank you. There's one more question. With the integration of StringDB, is the literature-based text mining annotated for interaction included, excluded?
Included or excluded?
Yeah, or
yeah. No, I'm not sure actually on the detail. I mean, we're aware that StringDB includes this literature mining process. I'm not sure exactly how we've chosen to do that, so I'll have to get back to you on that.
Okay, we'll take one more question and then we'll move on to Dr. Oprea's talk. So here's the last one is, if a biotech is looking for a target for a disease, can it find it in the top 10 list of the diseases from open targets?
If that's Pankaj from used to be at GSK. I would say yes, most of the time, one would hope, but for many genes, like for many diseases like IBD, there's hundreds of candidates. So I think what we think about of the Open Targets platform as is the starting point for your research that you want to do on this target, on the disease, to get the list of targets. I think you have to make a series of other filters to go further. One of the things that we don't include, which I think is very important, is novelty. I know Tudor has done some work on that, and it's something that we're interested in working on with him. Because we're not including anything about the commercial factors that are involved in making that choice. So I think there's lots of other things that you would want to consider. But in terms of the evidence with association with disease, I would hope that we do include, not necessarily in our top 10, depending on the disease, but in the top ranking.
Thank you. And on that note, I think we're going to turn it over to Dr. Tudor Oprea, who is the PI for the Knowledge Management Center for IDG, as well as co-PI for the RDOC. And he'll be talking about the Illuminating the Druggable Genome, Drugs, Targets, Phenotypes, and Diseases.
Hello, everyone, and thank you for attending this webinar series. I wish I had a picture of everyone that's involved in the IDG, the development of Pharos and Drug Central, etc. Probably before the pandemic, taking such a picture would have been possible, but I will make it my mission to accumulate as many screenshots as possible to compile that picture from a virtual perspective. So with that in mind, I am trying to give you a quick tour of what we're doing with two of the databases, Drug Central and TCRD, and also introduce to you the knowledge-based classification of proteins, which is what we call the target development levels, and then with some emphasis on diseases, rare diseases and cancers, try to illustrate how you can use the Pharos platform. So essentially, as we contemplate the use of informatics, machine learning, data science in drug discovery, there's sort of three angles one can consider: diseases, drugs, and targets. So we think of these as the three pillars of IDG, of drug discovery. And in principle, you can think of multiple ways and angles in which we can contribute. And specifically, I will focus on diseases and targets at this point in time. But before I do that, I want to highlight that the concept of annotating drugs to targets is a rather difficult task. This paper that is highlighted here, it took us about 7 years to do the update. So we manually had to go through over 1,500 drugs and almost 1,000 biomolecular targets. And even though this compilation is widely cited, we're still not 100% sure that it represents what people would call the absolute truth. And the reason for that is it relates to the difficulty of ascertaining that a specific target is actually how the drug works. The only way for that experiment to happen is, for example, if you were to CRISPR-Cas the target out, still have a disease model, add the drug, and to show that the drug does not have an effect. So those types of experiments are not trivial to do. We're, of course, hoping that as science progresses, we add more illumination to the process. A lot of that information that was captured in the Nature Drug Discovery 2017 paper is updated and maintained almost live. We run updates every 3 to 6 months in this platform called Drug Central. The most recent paper came out in Nucleic Acids Research. And just to highlight, we specifically try to illustrate from a chemical structure perspective, but also disease annotations, side effects mined from the Federal Adverse Events Reporting System, which is maintained by the FDA, as well as drug labels from DailyMed. We try to update a significant amount of information. And of course, one of the newest additions to this is a predictive platform, which we call Redial 2020. This is, of course, a pun, but essentially what we try to highlight is the possibility to use this platform to try to predict novel bioactivities for COVID-19. And even though this paper that describes Redial 2020 was accepted in principle in Nature Machine Intelligence, we're still not past that accepted tagline, mostly because papers, figures don't have the required DPIs, et cetera, et cetera. So every week we think the paper was going to be accepted and we still have to work on it. So with that rant out of the way, I just want to highlight that the IDG program is a really complex endeavor. The consortium has a number of centers, including the experimental groups that work on GPCRs. That's Brian Roth and Brian Choquette, and then Lily Jan and Mike McManus lead the ion channels effort, and Gary Johnson leads the kinases effort. There's an outreach effort led by Stefan Schurer, together with Larry Sklar and myself. Avi Mayan leads our complementary knowledge management center component out of Mount Sinai, and then there are several cutting-edge informatics technologies tools led by the investigators listed here. There are also a number of new additions, the experimental groups doing the R03 projects. There's, by my count, 32 of those. And so that would be too complex to list here. And similar to what Ian has described for the Open Targets platform, we have a rather complex pipeline for data acquisition and integration, in addition to data that's generated by our experimental groups, which goes straight into Target Central and Pharos. We, of course, curate the data from literature as much as possible. And by literature, we include a number of resources. We also include disease ontologies and tissue ontologies, etc., as well as chemical information coming from Campbell. This is not unlike what Ian was presenting. Plus, we mine information from patents and drug labels as much as possible. So all that goes into Target Central, which can then be interfaced via the knowledge access portal Pharos. There's, of course, a direct line to developing machine learning. So we have a GitHub repository where we maintain protein graph ML. So those of you who are interested in that, I will touch upon it very briefly today. And then we try to reevaluate and constantly understand how to annotate this information. So part of the integration process is making these databases talk to each other. For example, string was mentioned earlier. We maintained a string database or integrated the string database. We also integrate, for example, with viral protein-protein interactions through something called phipster. And I just realized that probably string is missing on this list, which was supposed to be alphabetical, so I'll have to correct that. We also keep track of information that comes from 3 other Common Fund projects. One is GTEx, the other one is the International Mouse Phenotype Consortium, and as well as the links cellular signatures. So all this information gets extracted and it gets into protein attributes, disease attributes, and ligand attributes. And that allows us to examine the human proteome and try to define the question, how much do we know about the human proteome? So we have proposed this knowledge-based classification where we consider mode of action drug targets get this so-called T-CLIN label that was subject to the NRDD 2017 paper. T-Chem are those that are annotated with chemical matter, and the bioactivities for the T-Chem which allow us to lead to that are illustrated here. So we look at the bioactivities of drugs for individual target classes. And it turns out that for kinases, the cutoff is at around 30 nanomolar, whereas for ion channels, the cutoff would be at about 10 or even almost 20 micromolar. So with this kind of dynamic range, when we don't reach that range, we just default at 1 micromolar for unknown activities. And in addition to that, we have 2 other categories, T-bio and T-dark. And T-dark are the proteins that are not very well described in the literature, whereas T-bio are proteins that lack either chemical probes or drugs, but we have a reasonably well understood biology. And the way we look at T-bio is that if, for example, you have a experimental evidence code according to the Gene Ontology Consortium, that would progress you to T-bio. We also look at the presence of annotations in OMIM, the Online Mendelian Inheritance in Man database, or passing out of these criteria that keep the protein into the T-dark designation. And specifically for T-dark, one of the main criteria is that we count fractionally the number of publications. Say, for example, you're looking at the insulin receptor. If you have 100 papers discussing the insulin receptor and only that, then that fractional count gets to be 100. But if 100 papers discuss 20,000 proteins, of which the insulin receptor is 1, then that number becomes very, very small. We also, in parallel, look at counting antibodies. we found there is an interesting relationship between publications and the number of antibodies available. However, it sort of illustrates that when you have molecular tools available, then you can describe a protein better. So that progresses a protein out of T-dark, but that is a somewhat independent number from Jensen Labs score, the fractional paper count. Also, we look at gene reference into functions. These are maintained by our colleagues at NCBI. So all this information is available in Pharos. This is a tool that's been developed by our partners at NIH NCATS, the National Center for Advancing Translational Sciences. And you can query the Pharos portal either by target. This is by far the most developed part at the moment, but we've made significant strides in improving the query by diseases and that annotation, as well as ligands. And by ligands, we mean ligands that have been previously processed in Campbell or in Drug Central. So the take-home message that I think I convinced you of is that there is a knowledge deficit when it comes to protein information. About 1/3, a bit less than 1/3 of the human proteome remains understudied, and maybe 3%, close to 3.2% is now currently targeted by drugs. So of course, if you want to work on dark genes, which is what we're interested in, that remains a high risk endeavor. So I'll try to illustrate that point. So recalling the fractional paper count, Lars Jensen maintains a resource called Diseases. And we're in the process of releasing a new publication which updates the Diseases resource. But essentially, there are three main channels of information in diseases: text mining, knowledge, and experiments. And when it comes to text mining, the database system has evolved significantly by looking not only at named entity recognition, but also including a bidirectional encoder called BioBERT for full text mining for that subset of Drug Central, not Drug Central, PubMed Central, that allows that type of research, and in addition to abstracts, of course. And on top of that, there is a experimental channel which currently includes the target illumination by GWAS Analytics, or TIGA, a tool that was developed in collaboration with our group. So Jeremy Yang was the lead on that, and that is a paper that's accepted with revision in Bioinformatics. So you can read about it hopefully in the next month or two. So when it comes to disease annotations, one of the topics that is rather disturbing is this translational gap, specifically for rare diseases. The number of publications per rare disease is 3 to 4 orders of magnitude higher than the number of drugs that are available. So if you look at orphan drugs, which have a rare disease designation, that's in the lower hundreds, whereas the number of publications is by now reaching millions. And part of the problem is, of course, our definition of rare diseases. So we struggle with this. And one of the recent efforts we did together with the Monarch team, led by Melissa Handel, was to ask the question, how many rare diseases are out there? And the answer, which is still often quoted as being 7,000, we think that comes from OMIM, except the data in OMIM is not strictly for rare diseases. But if you look at rare diseases, that number is probably by as much as 50% higher. So over 10,000 rare diseases out there. And you can read more about it in this commentary. So one question that we are trying to address is how much can we learn from phenotype mice coming from the International Mouse Phenotype Consortium. And based on a conversation we had with our colleagues from IMPC, with Jeremy Mason and Violeta Munoz Fuentes, we tried to address the question, how much do we know about the relationship between human genes and mouse phenotypes? So a rather recent mapping at the end of February, we concluded that when you look at early adult homozygote mice that have been phenotyped successfully through the pipeline at IMPC, and they have a very strict pipeline, that answer is about 7,000. And out of those 7,000, almost 4,000 are what would be considered understudied, as in having less than 50 fractional publications according to two different criteria, PubMed, which is Lars Jensen, and also PubTater. That we don't know. So it turns out that in the to be or not to be phenotyped question, one of the topics that one should consider is this fraction of about 1,400 mouse models that do not have significant phenotypes. And that definition comes from the fact that, for example, the black ones have at least 13 large set procedures. So each procedure may have multiple measurements that have been through the pipeline, but not significant compared to wild type. And then between 7 and 12 is a separate category, and then less than 6. So it turns out that about 800 of these, we would be confident to say that they lack significant associations. It turns out that if you query further into these subsets, you find that perhaps as many as 300 of these subsets are associated with rare diseases. And there is another slide, which I'm not showing in the interest of time, which shows that about 40 of these also have diagnostic value and are used in different gene diagnostic panels according to data from Orphanet. So I should have mentioned that all this data comes from Orphanet. So in short, perhaps as much as 20% of these mice models could probably serve as loss of function models that could be associated with human rare diseases. So the short version, the take-home message is that you could prioritize dark gene research using the IMPC. We think that the negatives that come from IMPC are true negatives. And the reason that statement is here has to do with the rigor of statistical measurements. So each of these associations has to be evaluated in a number of, according to a number of statistical parameters. And when the relationship is not available, then we can consider that to be a true negative. So when you think of monogenic rare diseases, which are associated with phenotyped mice, you could think of them as progressing straight to the relationship to a human disease. So with that in mind, perhaps as many as 300 mouse models could be available for rare disease research. So in the last vignette, I'm going to switch gears quickly to cancer. A lot of literature is dedicated to the subject, how many proteins are associated with cancer. The answer is perhaps as much as 2/3 of the human proteome in the primary literature has been associated one way or another with cancer. And out of these 14,000 proteins, almost all of TCLIN are included, and almost all of TChem are also included percentage-wise. But then the question is how many of these are really important in cancer? And Ian has already mentioned the Cosmic Cancer Gene Census. Version 92, which was released in January, has 739 genes. These are categorized in 3 categories: Hallmark cancer tier 1. Tier 1 also includes the Hallmark genes, and tier 2. And of course, tier 1, which includes Hallmark, is related to the strict evidence. So there is a clear causal relationship between these genes and cancer. Whereas with tier 2, the evidence is nearly there, but doesn't meet the exact requirements. And just as a vignette, you can, if you follow kinases, you will see that a significant number of kinases are present in TCLIN and TCAM throughout this upper layer of panels. So we asked the question, what's the relationship between these cancers? And we looked at the Cancer Cell Line Encyclopedia, which is a subset of the data that's integrated into TCRD-FAROS. And using this t-SNP plot, just looking at gene expression data, what you see is all of the blood cancers are sort of clustered on the right of this plot. And then the other cancers are in the rest of the plot. Some of them have very clear clusters. For example, male lung cancer is here, whereas female breast cancer is here. Whereas some others like pancreatic, esophageal, kidney, brain, they're sort of spread throughout. So it's not that trivial to establish the relationship. This nonetheless allows us to inform machine learning models. So the other thing that you can do, of course, is cluster by gene types. So this is the same data set, except instead of looking at the primary disease, we're looking at the genes and their designation in the biological terms. One of the most interesting clusters, which seems to be very focused, very narrowly focused, is olfactory GPCRs. They seem to play a special role. In particular, some of them that are overexpressed in a number of cancers are probably of interest and worth examining as potential drug targets. So what we did is we tried to develop machine learning models using an approach called MetaPath, which I won't go into, but I'll be happy to tell you in private or point you to some literature. And then we asked the question, can we build a model that uses the Hallmark cancer genes as positives? And then can we build a negative set that would serve as the counterpoint? And this counterpoint, of course, is somewhat artificial. So the definition of that was we wanted housekeeping genes that were not associated with cancer according to the diseases database. So they are roughly equivalent in numbers. So we think we developed a somewhat okay model. I won't go into the tier 1 and 2 models because we think that the Hallmark model is the most significant, but based on that type of information, these are 20 genes that are not included in tier 1 or tier 2, which are strongly associated with cancer. The ones highlighted in blue are of interest to the IDG consortium, so kinases and GPCRs. There is no ion channel in the top 30, but there are some ion channels which are associated. If you go in the list into the top 100 predicted genes, some of them are actually ion channels. And it turns out that for people working in cancer, most of these targets are very familiar. So we think that they might be considered hallmarks of cancer. So they probably require additional attention. So with that, I stop. I hope I convinced you that you can use dark genome research to benefit both rare and common diseases. Of course, the question is, can you use machine learning? The answer hopefully is yes. And I'll be happy to take questions. Thank you.
Thank you, Tudor. Feel free to type in questions in the Q&A below. Maybe you could address, you mentioned the machine learning aspects. So there are some work on having machine learning elements be available via FAROS. So for other issues of interest, so to speak, for people to do machine learning? Are those being facilitated?
Yes, so we're currently working with Avi Mayan and his team to develop a hypothesis page. We envision that within the next 3 to 6 months, we will have that possibility where you will be able to use a user input list of positive label, as it were, genes, where the positive label genes would be what you consider to be associated with the disease or phenotype of interest. And you would have to provide a list of negatives. And if you don't provide a list of negatives, we can provide that for you, except it's less, perhaps less relevant to the model in question. There is already a working version of this using Apiter, which is something that Avi Mayan and Alex Lachman have developed at Mount Sinai. We also have the MetaPath implementation in Python that's working with the GitHub associated with it. So hopefully we'll be able to roll this out shortly.
Thank you. There is one question that is saying, how are you, Dr. Oprea, how are you using machine learning?
So we are using machine learning two ways. One is to try to prioritize genes that are associated with a specific disease, whether it's Alzheimer or chronic pancreatitis. We try to ask the question, if you start with an input list of genes, what are additional genes that you should be looking at? The other question that it's related to this is, of all the genes that are associated, so you have, say, a list of input genes, say 50, and then another 50 that are predicted, can you combine them, for example, with the string interaction network and visualize them in string and try to figure out which ones you could modulate to have a better clinical outcome. So that's how we use the machine learning component.
Thank you. There's also a question saying, how useful is a model that separates Hallmark cancer genes from housekeeping genes for predicting cancer targets?
So in this specific case, as I think I pointed out, to date, probably every single protein that's in the human body has somewhat been associated with cancer. But even though 14,000 of these genes have literature, there's only 300 that are considered hallmarks. So clearly the bar for hallmark is much higher. So what I try to do is build a model to ask which additional genes are hallmark-like. And we, of course, think of housekeeping genes as being important and essential to cell proliferation and division. So they might be playing a role in cancer as well. And I'm not saying they don't, except out of the 3,000 housekeeping genes, I selected less than 300 to be considered as negatives. I'm not sure they're true negatives, but there's a decent chance that they are negative, since nobody has associated them with cancer in the last, I don't know, 30 to 50 years. So the top genes that are predicted, people who work in oncology tell me that they are reasonably good assumptions to be hallmark-like. So that's why we're trying to build that model.
Thank you. I have one more last question here, and it says, do you anticipate any challenges in translating cell line or gene data between mouse and human? There seems to be a gap here that leads to problems with murine cancer models.
That is correct. There are challenges. We are working on developing this. In some cases, the phenotypes translate rather well. So, for example, we examined data related to 5 different mouse phenotype models related to glucose levels and insulin levels, and they seem to translate rather well in the field of type 2 diabetes. The other project that we looked at was the relationship with cardiac phenotypes. So IMPC has an excellent pipeline to look at ECG and echocardiogram changes. And if you look at those phenotype changes, there are very clear relationships. The ECG ones relate, of course, to cardiac physiology, and the echocardiogram relate more to cardiac malfunctions. So there's like different relationships there, but those relationships do exist. So we're now trying to put that foray into rare diseases with the hypothesis that even though IMPC has this 540 set of experimental values they look at, obviously they cannot capture everything that you see in, say, a human rare disease. So then the question is, can we see what they missed, and ask the question, is that change of that particular metabolite or maybe that particular change in phenotype something that you overlooked when you looked at that mouse model? So that's the plan with the rare diseases. So clearly it's not a one-to-one relationship, but that relationship is present and there is signal in that, which is valuable.
Great, wonderful. Well, we've come to the time, and I think this has been a wonderful inaugural session for our e-Symposium. And I would encourage you all to check out the fact that we're going to have a next one, actually. And let me click and talk at the same time, which happens on March 30th, and the theme is on kinases, and we encourage you to sign up and join us then. We'll have two speakers, Dr. Martin Kolkwitsky and Dr. Gary Johnson. And thank you again to today's speakers, Dr. Dunham and Dr. Oprea. And if you have any further questions or issues you want to email, feel free to email the idg.rdoc@gmail.com, and I will pass on questions to the speakers if need be. Thanks again for joining.
Thank you, everyone.
Thank you.